In this video, I'm going to show you how to import data from your DNAnexus account into GenePool. I'll start by actually showing you my account in DNAnexus at https://platform.dnanexus.com. I've already logged in, and here you can see all of my projects. In particular, an RNA-seq project and an exome project. In each of these projects, I've actually already run pipelines culminating in files that I'd like to import into GenePool. In my RNA-seq project, I have BAM files that have been indexed for 6 samples. And in this exome project, I have VCFs as well as BAMs that have been generated for 11 samples. So what I'm going to do is actually go back to my GenePool login page and log into my GenePool project account. The first thing you're going to need to do is make sure that your GenePool account is connected to your DNAnexus account. In order to check on this, you can click on the Account tab at the top right of GenePool, go to Connected Apps, and you can see here my account already happens to be connected. If your account is not already connected, you're going to want to go through the workflow. I'll recreate that by clicking on Reconnect. By clicking on Reconnect, I'm asked to authenticate and log into my DNAnexus account, which I am doing so. And after doing so, you can see that I'm back in my GenePool account, and now I'm connected. And now I can go through my normal import workflow. For the RNA-seq project as an example, I'm going to name the project, walk down the gene expression workflow, ask GenePool to actually compute counts from the BAM files that were generated using an RNA-seq pipeline on DNAnexus. And I'm going to ask it to compute counts from non-directionally prepped FASTQ generated data from an Illumina sequencer. And in the next step, I'm asked to select an external source. So in this case, I'm going to select DNAnexus. And since I'm already authenticated, you will see that GenePool will render the tree structure that is in my DNAnexus account and show me the projects that are available. So here you can see the RNA-seq project and the exome project, as well as the other ones that are in my DNAnexus account. I'm going to click on the RNA-seq project and select those BAM files that you saw in my account. It's important that these BAM files actually be indexed when you select them. So for example, if I were to select a sample that is not indexed, you would receive an error like this one. So I'm walking through my 6 samples and selecting all 6 of them. And once I've selected them, I can click Next Step. You'll see all 6 of them are listed here. And if I wanted to actually import these samples, I can click Submit Project, Submit Project Import Job, and GenePool will go off and pull those BAM files and import them into GenePool. I'm not going to actually submit that job right now because I've already imported that project, but I will walk you through a similar workflow for bringing the exome data. So again, click Import Data. I'm going to now walk through the sequence variation workflow so that I bring in the VCFs, the sequence variation data. Click Next. In this case, I can actually import VCFs from my desktop or from an external source. And I'm going to select DNAnexus as an external source. And again, you will see the tree structure render that represents my DNAnexus account. And now I'm going to select the exome project, and again click through these samples and select the VCFs. So in this case, I'm selecting 11 VCFs for this particular SRA project. So these VCFs were again generated from a DNAnexus pipeline, taking the FASTQ files as input and running it through a BWA GATK pipeline, culminating in 11 VCFs for these 11 samples. And now that I've selected all of them, I'm going to go to next step. You can see all of these 11 samples are ready to go. And again, I'm not going to click on submit project since I've already imported this data, but if you need to import the data from DNAnexus, all you would need to do is click submit. And once your job is completed, which you can track in the job monitor up under the admin menu, you will get an email stating whether or not your data has successfully imported. And I'm going to show you in my projects area where I've imported this data. So the RNA-seq project was imported earlier today. And when this data is first imported, it would actually look a lot like this without any clinical metadata associated with it. So in order to get these columns of metadata that I've imported, that I've attached to these samples, all you need to do is right-click on the project or click on the gear and go through the Update Clinical Data workflow. And if you'd like to attach BAM files for use in the genome browser, you can do so using the Update BAM paths. Same goes for the exome project. We have videos to go through each of these workflows if you need more details.